I'll be starting us off today. Thanks, Adita, for that introduction. My name's Shayna, and I'm joined by my capstone team today, Amena and Kelly, and we'll be discussing our capstone, digitizing post-CAR T-cell therapy patient monitoring. And thanks again to everyone for being here today. So to start off, I'll discuss some background information. When setting out to do this capstone project, our team joined together from different backgrounds, but with a common goal to do a project in cell and gene therapy that would have a real impact. And cell and gene therapy is the transfer of functional genes and/or cells to restore a relevant function within a patient. We quickly narrowed in on chimeric antigen receptor, or CAR T-cell therapy, because it's the most well-established cell and gene therapy in Canada, and we also had connections in this area to help us get started. CAR T-cell therapy is a cellular treatment for blood cancers, including specific types of lymphoma and leukemia. When a patient has already undergone the standard treatment and nothing proves to be effective, they previously might have had no other options. But in 2018, Health Canada approved the first CAR T-cell therapy product, which is a potentially life-saving treatment for this patient. CAR harnesses the innate power of the human immune system using genetic engineering to reprogram a patient's own immune cells, called T cells, to target and kill the cancer cells in their body. To receive this one-time treatment, a patient's own blood containing T cells will be collected in the hospital and then sent to a specialized lab for genetic engineering and growth in cell culture, and then finally reinfused back into the patient where the CAR T cells live, grow, and kill the cancer. After narrowing in on CAR T-cell therapy, we first conducted preliminary discovery and had conversations with people working at the different centers providing CAR T in Ontario, as well as members of industry, pharmaceutical companies, health economists, and different academic researchers. We discovered that there are significant problems related to the need to monitor for adverse effects after a patient receives CAR T-cell therapy. And the two main adverse effects are cytokine release syndrome, or CRS, and immune effector cell-associated neurotoxicity syndrome, or ICANS. CRS is an inflammatory response caused by the CAR T cells themselves. So when the CAR T cells detect a cancer cell, they become activated and start to produce inflammatory markers called cytokines. And this can occur in a positive feedback loop, resulting in a cytokine storm that can cause fever, hypoxia, potentially multi-organ dysfunction if not treated in a timely manner. And severe CRS occurs in up to 46% of patients within 7 days of infusion. ICANS is a specific type of neurotoxicity that can cause tremors, memory impairment, and other neurocognitive symptoms. It's usually reversible with proper treatment and occurs through a similar mechanism as CRS because the CAR T cells, other immune cells, and cytokines can all cross the blood-brain barrier and cause an inflammatory response within the central nervous system. ICANS occurs in 20 to 70% of patients within 10 days of infusion. Currently, in order to monitor for these complex different adverse effects, patients have to stay in the hospital for up to 40 days after receiving their CAR T cells, and they undergo intensive monitoring while they're there, including vitals and temperature checks every few hours to monitor for CRS, and cognitive function tests called the ICE assessment multiple times a day to monitor for ICANS. After patients leave the hospital, they often have to stay in outpatient centers or hotels nearby so they can return frequently for appointments or in an emergency. The treatment itself is already quite costly, and the current monitoring protocols significantly add to that cost. These high costs affect the healthcare system, who covers the cost of care in Canada, but also patients and families who are responsible for costs of commuting to and from the hospital, long-term stays, and the cost of having a mandatory caregiver who must also take time off work. This impacts the quality of life of the patients and also the healthcare system and pharmaceutical companies who are facing capacity issues already, and these are predicted to get worse as more CARs are approved. Through outreach and literature review, we found that there are clear gaps in these current post-CAR T-cell therapy monitoring practices, namely the extended hospitalization and travel time, lack of remote monitoring, which is being implemented as a solution elsewhere, but not yet in Canada, and the lack of standardized and long-term data collection across centers, all of which are costly and time-consuming for the healthcare system, patients and families, and contribute to the capacity issues that we're already facing. So with a new understanding of these potential problems in the field to address, we now had to work to narrow our scope, and we drew from the Toronto Translational Framework to specify what unmet need to focus on through this project and brainstorm a testable solution that we can continuously verify throughout the project. It was at this point that we also met our industry partner, CureTracks, who was developing a remote monitoring solution in the allogeneic stem cell transplant space, which has a similar patient population to those who receive CAR T. We also continued to speak to others in the field and confirmed that remote monitoring may in fact be a feasible solution. That being said, we decided to focus on addressing the unmet need for a tool to remotely monitor CAR patients in Canada. We understood that this was a big problem and that there might be many steps to tackle this. So we narrowed our scope further to create a feasible project within our timeline and focused on step one, which was creating the tool that might be used in the future to remotely monitor patients. We then designed a study around the research question, which parameters, functions, and design features should be included in a tool to provide effective remote monitoring and standardized data collection for CAR T-cell therapy patients in Canada. And to answer this question, the project objectives included gathering input on capabilities and user experience by engaging healthcare providers, and then using this engagement to inform the design of a remote monitoring tool, which would then be prototyped with the goal of improving patient outcomes and quality of life. So now we'll go through a few of our methods overview. So we understood that a tool for healthcare providers to remotely monitor CAR patients would have two different interfaces and two different end user groups. One would be the healthcare providers, and two would be the patient and caregiver group. So to address this research question, we adopted a human user-centric approach, and we wanted to involve both of these end user groups from the start. So we plan to conduct semi-structured interviews with 10 healthcare providers and 10 patient caregivers. Interview transcripts were to be coded and in vivo to identify and categorize themes in the data, and any quantitative data was analyzed in Microsoft Excel. The final recommendations would be implemented into a prototype of the tool. We recruited participants through various organizations such as Leukemia and Lymphoma Society of Canada and Cell Therapy Transplant Canada that fit the inclusion criteria that are listed on the screen. And now I will be passing it over to Kelly to discuss the results.
Hi, everyone. My name is Chimaobi, but I go by Kelly, and I'll be telling you about the results of our study. So the first thing we did was to conduct a literature review. This gave us insight into the current monitoring protocols and also allowed us to compile a list of parameters that were key indicators of CRS and ICANS. Through literature review, we found that some key physiological symptoms for CRS included a fever, hypoxia, hypotension, and tachycardia, and these are currently being measured with thermometers, pulse oximeters, blood pressure monitors, and documented using analog methods. On the other hand, neurocognitive symptoms associated with ICANS are measured using a simple cognitive test. The literature helped us create a list of questions, and these interview questionnaires were then presented to the study participants. We interviewed 10 healthcare providers within the CAR T space, and this included physicians, nurses, pharmacists, and social workers. We asked them a range of questions, all related to post-CAR T monitoring and follow-ups. The healthcare providers answered these questions based on their knowledge, their expertise, and also what is currently being done at their respective centers and how it could be possibly applied to remote monitoring. So let's get into some specific results. So the first main question we asked the participants was to prioritize a list of parameters and tell us what is most important to monitor for adverse side effects. So here on the x-axis, we have the different parameters, and on the y-axis, we have the number of participants that categorize them as low, medium, or high priority. So if a parameter was ranked as medium or high by majority of the participants, then it would be included in the monitoring tool. And based on our interviews, majority of participants categorized temperature, oxygen saturation, blood pressure, and the I-score as high priority parameters. Additionally, we also wanted to know the minimum frequency at which each of these parameters should be monitored. And similarly, the frequency that was selected by majority of the participants was also included in the tool. It was recommended that temperature, oxygen saturation, blood pressure, and I-score be monitored 4 times a day, while self-reported parameters like motor findings, headaches, muscle aches, and pain be reported 3 times a day. With regards to the static parameters, these are the one-time entry data points that are part of the initial setup of the tool. So they would include biological sex, cancer type, comorbidity, CAR T cell therapy products, and these were all categorized as high priority by the participants. They also had comments on when and how they wanted the information to be displayed. Participants suggested including graphs to longitudinally show display trends over time. And majority of participants also stated that they are willing to dedicate less than 30 minutes a day per patient for remote monitoring. So there are some additional features that healthcare providers wanted to see, such as an option for the patient to specify if blood pressure information was collected standing or sitting, because this gives insight to postural hypertension that occurs in some CAR patients. Another interesting novel find was that one healthcare provider said that they had noticed that changes in sleep patterns had been correlated to the onset of ICANS, and this was not mentioned anywhere in the literature. Additional features were more patient-reported symptoms like lightheadedness, severe or sudden headaches, urine output, bowel movements, and shortness of breath. Another healthcare provider did emphasize the importance of collecting ethnicity data because this facilitates research into the racial differences within the CAR T space. So we plan to interview 10 people with lived experience, including patients and caregivers. Recruitment methods included outreach to patient groups like the CTTC and Leukemia and Lymphoma Society of Canada. In the end, we were not able to recruit patients and caregivers to provide input for this iteration of the study due to the small population and the limited timeline for the capstone. But as an alternative to collect general feedback, we conducted a pilot usability study. So we performed a pilot phase for a formative usability assessment. Now, I know it's a mouthful, but all it was a simple assessment where the participants, including peers and colleagues that were not directly involved in the project, provided feedback on the design, usability, flow, navigation, and accessibility of the remote monitoring platform. And based on the results and recommendations of our interviews, the literature review, our outreach, and also the formative assessment, we decided to prototype a mobile phone application and a web dashboard. Using Microsoft Visio, we created a process map to show the flow of information across various parts of the platform. Now, this platform consists of 3 main components. There's going to be the patient's app, which will primarily be used by patients and caregivers. We're also going to have the physician dashboard that will be accessed by members of the health team, including nurses, physician assistants, and doctors. And we also have a group of integrated devices. To start, respiration rate, heart rate, sleep, and physical activity will be monitored using a smartwatch. All the data from it is automatically connected to the mobile phone application and will then be automatically uploaded to the physician dashboard. Temperature will be measured using a standard thermometer, and the value will be manually inputted by the patient. Oxygen saturation measured with a pulse oximeter and also reported. Same with blood pressure, which will also be manually measured and reported by the patient. All these parameters will be reported into the patient's app multiple times a day or as directed, and the patient is also required to enter other self-reported symptoms like headaches, pain, fluid intake, and so on. And then all the information from the app would then be on the physician dashboard where the healthcare providers are able to monitor the patient in real time. So I'm going to pass it over to Amena now to talk a little bit more about the results of the formative assessment I had mentioned earlier.
Thank you so much. My name is Amena and I'll be taking over. So as Kelly had previously mentioned, participants not directly involved in the study provided feedback on various elements of the prototype of the mobile phone app. Participants stated that the overall layout of the app and general navigation of the application is straightforward. On the other hand, participants stated that instructions on some of the cognitive assessments were unclear, and it was observed that users would perform the tasks incorrectly multiple times, and it was only correctly performed when an example is shown. Adjustments were made to the final prototype based on the feedback. Figma was used to prototype the mobile phone application and the web-based dashboard. The mobile phone application is how patients are able to input their vital and daily symptom monitoring for their physicians to monitor in real time. and recommendations from healthcare providers were integrated throughout the app to determine which vitals to monitor and the frequency of each parameter. The web-based dashboard is for healthcare providers to be able to access how the patient is doing at a glance. We incorporated their recommendation and have majority of the vitals and reported information displayed throughout graphs that allow them to view changes over time. The most important static parameters are also displayed on the left. Preliminary development of the app and web-based dashboard was done in collaboration with the TechTIS programming team. An architecture of CureTrack's existing monitoring platform was maintained and redesigned to monitor for CAR T-cell therapy patients. Now we will be talking about the value proposition, limitations, and future work that could be done for and using this platform. To begin, an important part of the study was digitizing the cognitive assessment used to assess ICANS. For example, the treatment facility parameters are asked, in the treatment facility, participants are asked to write standard sentence on a piece of paper to assess motor impairment. With our app, we have the patients take a picture of a written sentence or use a stylus with a scribble tool. Patients are also asked to name 3 random objects in the room as part of the ICE assessment. In the app, we have them play an audio file and select the corresponding picture. The formative assessment was very useful here because we got feedback on making the objects universally recognizable unless there's a case of cognitive impairment. Digitizing outpatient CAR T monitoring will be valuable to many stakeholders. The primary beneficiaries of the intervention are CAR T patients and caregivers. This intervention provides the opportunity to reduce costs and improve patient experiences. Within the CAR T space, hospitals are currently offering CAR T to adults in Canada will benefit from remote monitoring because it will allow limited hospital resources to be redirected and help increase capacity. Other major stakeholders include the drug companies that manufacture CAR T that have already established that they're interested in long-term data on CAR patients. Some limitations to note is that we had a small number of participants involved in the study. This iteration of the remote monitoring tool was designed without input from people with lived experience, and due to our limited timeline for this phase of the project, the scope was narrowed to focus on the clinical framework of the app, so feedback from healthcare providers was prioritized. As well as the platform still needs to be validated to demonstrate efficacy and safety, as well as validation of any enabling technologies that are included. The next phase would involve collecting patient and caregiver feedback, and additional future studies may include a feasibility study to assess the practicality of the remote monitoring platform. Endpoints may include reporting adherence, time burden, user retention, and user satisfaction. And finally, a validation study will be required to assess efficacy and safety of the platform. Endpoints to demonstrate efficacy, safety, and value may include time spent monitoring by healthcare providers, patient and caregiver satisfaction and quality of life, cost of treatment, and patient outcomes.
To conclude, there is currently no comparable remote monitoring tool available at any of the CAR T centers in Canada. The current monitoring practices are mostly paper-based, center-specific, and not standardized. This innovation has the potential to improve quality of life, documentation of adverse effects, reduce costs, and connect centers across Canada by providing a tool to collect structured data that will eventually help to improve care.
So before we get to the Q&A portion of the defense, we do invite you to scan this QR code. This will give you access to a clickable prototype of the mobile phone application. I will also place a link in chat. We currently do have a functioning version of the app that's connected to the web dashboard, but it is still in test flight, so it's not available to download in the App Store. But this is the next best thing. So this is going to take you to a Microsoft PowerPoint presentation. All you need to do is put it into presentation mode and just click through it as you would the app.
And we would like to acknowledge our industry partner, Dr. Armin Gerbitz. We also want to thank our supervisors, Dr. Joseph Behrenbach and Dr. Edita Marcone, and the members of our project advisory committee, Dr. Brent Zanke, Eric Hammond, Dr. Paul Eisen, and Dr. Keith Party for all their support and guidance throughout this project. In addition, we want to thank MyTax for funding our capstone and giving us the opportunity to contribute into that cell and gene therapy space in this way. Thank you so much, and we welcome any questions.